Welcome back to vaccine design course. So here we did the homology modeling by automated modeling. Now the next step that we are going to do is alignment modeling. So for alignment modeling, the first step is to retrieve the protein sequence or the target sequence which we already have. Next, our step is to retrieve the template sequence. Okay. So in automated modeling, the template sequence they already automatically select and then do the alignment. Here in alignment modeling, we have to retrieve it manually. So for that, you need to go for protein blast. Either go to this particular link or search for protein blast NCBI. So search NCBI protein blast and go to the first link. Now, so this is the protein blast page where you need to enter your or paste your protein FASTA sequence which we already have. So I'm gonna copy and then paste it here. And next step that we have to do was to change the database from non-redundant to protein data bank or PDB, right? And then after that, the algorithm has already been selected as BLAST-P. Then click if you want to show the result in new window and click the blast button. Okay. So the steps that we did until this is alignment modeling where we did the protein blast. Now next is about the result. Okay. Now you can see the result. When you scroll down in the descriptions, you can see that the data or the result is being arranged based on the e-value. So our focus is on percentage identity, right? So let's arrange it based on the percentage identity. Okay, so let's click on percentage identity, and after that what we need to do is to click on the first accession number, which we have already done in our previous session, right? Yeah, so click on the accession number. Then our next step was we need to click on the FASTA click on the FASTA. So this one is the best template protein sequence, right? 4xAKA. Now you need to copy it with the FASTA header. So here for the previous lecture, what we did was we took the PDB ID and the chain ID, right? Now here you need to copy its FASTA header and the FASTA sequence, Control C, and then paste it in your notepad. Okay, so let's paste it in the existing notepad. Okay, or you can take a fresh new notepad and then save it as template protein. But let me just make a heading here as template protein. So this was our target protein, right? So target protein, you can save it in your own way. So I'm gonna rename this as proteins. That's about it. Okay, since I can get in the notepad all the information when I open it, so that's why I'm saving it in the same notepad. Okay, so this was target protein 1 and this was template protein 1. Okay, so we have pasted its FASTA sequence. Now we have both the sequence, the target as well as template. Our next step is to do the sequence alignment. Okay, so let me just show you the PowerPoint slide. So till this we saved the template protein. Now to do the sequence alignment, the another server that we have is the Clustal Omega. Okay, so let's go to this particular link or search for Clustal Omega. Okay, so I've searched Clustal Omega. Now click on the first result. So this is the home page of Clustal Omega where you can see the multiple sequence alignment where you can do it. So now enter or paste a set of protein, DNA, RNA are the options. So we should select protein because it's the protein that we want. Now you need to copy the sequence in any supported format, right? So along with the FASTA header, you copy your template protein and then paste it here. Okay, so you need to have your FASTA header, right? Why? Because if it's without header, then server will take both of them as one. So we have pasted the template sequence, but before that what you need to do is you need to paste your template sequence as well. So that's why FASTA header is very important. So above it, let's paste our target protein 1. Okay, I'm gonna copy it and paste it here. Then after that you need to press enter. Okay, so that you can differentiate both the target and template. Next step is to set your parameters, the output format. Okay, you can take any one of your formats. You can go to the help and documentation to see. Anyway, let's go with the default option, and then if you want to notified or be notified by email, you can do that, or you can just press the submit button and wait for the results. Now you have your results where you can see the alignment, then you have the result summary, phylogenetic tree, result viewers and others. So let's look at alignments now. Okay, not so you can see various alignments, right? So you can download this alignment by clicking the download alignment file. Okay, so click it. Now what you need to do is you need to copy this whole thing. Okay, copy this alignment and paste it in your notepad. So I'm gonna copy the whole thing, Control C, and again pasting it in the same notepad. Okay, so that I know that the first one is my target protein, second is template protein, and the third one was the multiple sequence alignment using both these target as well as the template protein. Okay, now what you need to do is just copy only this multiple sequence alignment file and paste it in a separate notepad, because we want to save it in a different format. Okay, so let's copy this particular thing. Okay, Control C and paste it in a new notepad, and then file, save as, and make it as .aln. Okay, so in your folder, make it as target template .aln. So we want to convert this as ALN. Okay, go for write .aln, save as type, go for all files, and then save. Okay, so now when you look in your folder, you can see the target template in ALN format, right? So this is an ALN file. Now, so we have an ALN file. This we need for separate procedure. Now let's go for, so what we have right now is the sequence alignment, right? Now next go for Swiss model. Okay, go to Swiss model and then let's start modeling. Okay, so now go search for Swiss model. Okay, then start modeling. Then in the modeling button, go for alignment mode. It's not automated modeling anymore that we are going to do. We are going to do the alignment mode, right? So click for the alignment mode. Let me just do it again. Okay, so that I can make you understand better. So go for Swiss model, the home page of it, and then in the modeling, go for the second result, which is alignment mode. So this is now in the form of alignment modeling. The next procedure that you need to do is you need to upload the target template alignment file. That means you need to upload the ALN file, which we have already saved it, right? Now upload it. So press the upload button. Go for Bioinformatics. That was the folder, and then upload the target template alignment file. So here you can see the two models, which means it says that for the uploaded target template alignment, 2 bio units or chains were found. So that's why we have 2 models now. Now let's take the first model and click on the build model button. Okay. So when you do that, you'll get the results, where in the results it will show the stability as well as the model template alignment. So the result is ready. You can see your model here. As you can see, there is sequence identity which is 100% and you have full coverage. So this shows the stability, and then you have the model template alignment, right? So click on that to see the model template alignment. So here we have to use this model for antigen antibody docking. So either use this model for antigen antibody docking, or you can go for automated model as well. So do the automated as well as the alignment model and then compare it with the result. Okay, so now what we have is antigenic protein sequence, right? Sequence. So functional, so till before that what we have used the term was protein structure, right? Now we have the antigenic protein sequence, or a model, not sequence, I would say antigenic protein model, okay, with the target and template. Now our next step, okay, let me just show you the PowerPoint slide. Our next step is to find the stability of this particular antigenic protein model. So to check the stability of it, why? Because protein instability can lead to accumulation of misfolded and non-functional polypeptide, which then chews up the proteins and splits them out as small fragments of amino acids. So to check, you can separately go to another server called as Molprobity. Okay, so go to this particular link or search for Molprobity. So search for Molprobity and click on the first result. Even though it says it's not available, let's see, it was actually available. So here in the Molprobity page, what you need to do is you need to upload the PDB file. So we did not download it, right? So this was the Swiss model result that we got, the model that we got. So what you need to do is click on model 1 and PDB format. Okay, click on PDB format, where you will get your PDB folder. Okay, sorry, in PDB format. So let's just rename it and keep it in your particular folder. Okay, rename it as model 1 alignment modeling. Okay, and then paste it in your particular folder. Okay, so I have pasted it. Then go to Molprobity, choose the file, which is again the model 1 alignment modeling, and then you need to click upload. Okay, click the upload button. So now the job is running. So they are checking all the PDB files, and then they have mentioned this is your PDB file and other information where the number of residues in the chain is mentioned. Click the continue button. Okay, now next is, these are the different options that you need. Okay, so here what we need is, we need to analyze the geometry without all atom contacts, because our aim is to check the stability of it, right? You can also add, hydrogen bonds, van der Waals contact and everything. Anyway, let's click on analyze geometry, and then after doing that, they have by default selected all the protein options, Ramachandran plots and everything. If you want, you can select the hydrogen bond, van der Waals contact, and then after selecting that, okay, scroll down and click run programs to perform these analysis. Okay, press OK button. So yeah, so the model that we used was from the alignment modeling. Okay, your exercise is to upload the file that we have got through automated modeling and do all these procedures. Okay, so now the results are ready. Here, look the Ramachandran plot values. Okay, you have to look the Ramachandran plot values, and if it meets the goals, then it means the antigen model is stable. So it says that it's 196 Ramachandran outliers, there are no outliers, so happy. Then favored, it's 196, 95.15. The goal is to be greater than 98. Since it's 95, it's okay. Okay, so look at the goal side by saying how much it should be needed, and see if the Ramachandran plot is favored or not. Okay, if it's favored, then it means that the protein or the model is stable. Then you can also view the multi-criterion visualization and the single criterion visualization as well. Okay, so this is how you need to do the modeling and to check the stability of it. Okay, so hope you understood this lecture. Thank you.